Okay, shall we start again, guys? Okay, let's get started. All right, so these are the various proteins like we saw the summary earlier. And what we are going to do is we are going to look at the replication and how some of these proteins play a role in that, okay? So first things first, remember the sequences I was talking about, the uni 12 and uni 13. They're called uni because they are universal primers. They're also self-complementary as well as highly conserved sequences, okay? The NCR, the SS-NCR is the segment-specific non-coding region. So what that means is each of these genomic fragments, they have their own non-coding regions. So it's highly segment-specific, okay? Now, let's see how these genomic fragments work and how the influenza virus actually replicates, okay? Because the mechanism is a bit more complicated than what we are used to seeing. So we'll go over that step by step. All right, so your fragments 1 to 6, they encode for a single protein, okay? 3 of them are RNA polymerase subunits. So one is called PA, PB1, and PB2. So these are the 3 RNA polymerase subunits that are encoded by these fragments. It also encodes the enveloped glycoprotein HA as well as neuraminidase that we have seen before. Now, in some influenza viruses, there's a second reading frame on segment 2. This second reading frame encodes for a shorter protein, which is called PB1-F2. This actually localizes to mitochondria, okay? And it can enhance the apoptosis of the cell, sorry. So remember, apoptosis does involve mitochondria. So this protein can trigger apoptosis or enhance apoptosis in these cells. And the alternate splicing can occur in the mRNA that's also transcribed from segments 7 and 8. And as a result, it can make different proteins. So for segment 7, you can either make M1, which is the matrix protein, or M2, which is an envelope protein. M2 is actually an ion channel, okay? And this is something that can be targeted for therapy, as we'll see later on. Your segment 8 can make NS1 or NS2, so non-structural proteins. So how do these segments work? Let's have a look. So segment 4 encodes for HA. 5 is NP, which is a nucleocapsid protein, okay? This is what encapsulates the viral RNA and control the functions in RNA synthesis. Then you have your HA here, which is hemagglutinin. This is the same hemagglutinin that we have seen before. So again, this is a major surface glycoprotein for the viruses, binds to the receptors, and results in membrane fusion at low pH. And this is also an antigenic determinant for this virus. And we'll come back to that in a bit. Then you have your neuraminidase, which is again a major surface glycoprotein. And this can actually result in destruction of the receptors. It can result in dissociation of viral aggregates and also is an antigenic determinant. You have your M1, which is the matrix protein, interacts with the envelope as well, nucleocapsid, as well as NS2. Lastly, you have your M2, which is an integral membrane protein, and it has the ion channel activity that's essential for uncoating and maturation of the virus, okay? And lastly, NS2, which is the non-structural protein, and this is something that directs the nuclear export of viral nucleocapsids. So what we'll see is that material moves in and out of the nucleus to actually replicate this virus, okay? And this is also interacts with the matrix protein. Your viral HA binds to the cell surface receptors and mediates the fusion with endosome, for example. And remember, HA binds to sialic acid residues, okay? And those sialic acid residues are present on the cell surface or cell surface receptors of various types of cells. So a lot of these are mucoproteins that this HA will bind to. So remember, HA can agglutinate RBCs because RBCs have lots and lots of sialic acid, and this is the basis for hemagglutination assay. The protein itself is a type 1 transmembrane protein. You guys have seen all this before. And lastly, it forms trimers on virus surface and mediates the fusion of the viral envelope with the endosomal membrane. So again, if you look at the figure, here's your HA, you can see the trimer, here's your neuraminidase, and your various genomic fragments present as these nucleoproteins, okay? And within that, you'll see ion channel, you have your RNA polymerase complex made up of PA, PB1, and PB2, your matrix proteins, and your non-structural proteins. So basically, it shows you all the proteins that we talked about. So how does fusion work? Remember, your N-terminal will get exposed to outside of the virion, and it has a hydrophobic C-terminal domain that inserts itself into the membrane. So it has a transmembrane hydrophobic domain near the C-terminal. And fusion is facilitated by cellular proteases. So for example, furin or trypsin can cleave this protein and release the fusogen, or release the fusion peptide, sorry, okay? And you can also undergo conformation changes after acidification in the endosome. So this is something we have seen before. It's the same HA protein, okay? Now the cellular proteases can cleave the HA into two subunits, HA1, which is the surface subunits, which is shown in blue here. That binds to sialic acid, okay? Your HA2 is the green fragment here, which is anchored into the viral envelope, and it has the hydrophobic fusion peptide that you can see here. So what will happen? Once the right trigger comes in, the conformation will change. It will insert itself to the target membrane and fuse the membranes, okay? Now, that's one way, that's cleavage by the proteases, but you can also change the conformation after acidification. So remember the M2 protein we were talking about? It's an ion channel. The M2 forms an ion channel, and it releases or facilitates the release of nucleocapsid from the virion. And the way that works is basically it allows acidification to happen. It allows the protons to enter the virus interior, so it acidifies the virus itself, and as a result, it weakens the interaction of your matrix protein with the rest of the nucleocapsids. So as the virus is getting acidified, the interior of the virus is getting acidified, you are going to start weakening those interaction of matrix as well as the nucleocapsids, okay? And as a result, this is a very good target for antiviral drugs. So one of the drugs, the amantadine, this is used in extreme cases to actually inhibit this M2, okay? But problem is, there's an issue of drug resistance because these viruses mutate fairly fast, and as a result, they can become resistant to these antiviral drugs. So let's look at the figure. Here's your, again, binding and internalization of the particle. M2 opens, and you have your influx of protons, and in this case, potassium as well. So this is early endosome at a pH of 6.5 to 6. And as it gets more and more acidic, your core will loosen, and eventually, when the pH gets to about 6 to 5, a little bit below 6, your fusion will start to happen. And at that point, your M1 will uncoat, and your nucleocapsid is now released into the cytosol, where it needs to get sent to the nucleus. Okay? So even though this is an RNA virus, it needs to get into the nucleus for it to work correctly and to replicate. Okay? So just to show you guys, M2 is a relatively small protein, so it's only about 97 amino acids, but it does form a tetramer. And as it does that, it creates a small pore in the viral envelope. So here you can see the C-terminal base, and here's the N-terminal region. Okay? Now, one thing to keep in mind here is it has that external N-terminal domain right here, and a transmembrane domain that goes in the membrane. Sorry, let's go back for a second. This is the transmembrane domain, and then it has a base that's much larger, and it's internal. Okay, rest of the protein will be up here. Now, the way it works is these two residues, or these three residues, play a major important role in opening of this channel. Okay, so tryptophan, which is W41, so tryptophan at position 41, this gate through the channel is actually locked. And the way it locks is that it interacts with another residue, which is aspartate at position 44. Okay? And as the protons come in, this particular histidine at position 37 gets protonated. Okay? So as the pH drops, histidine residue gets protonated. This causes a conformation change, and now this will open the gate. And as the gate opens, you can conduct protons across that channel. Okay? So again, here's your HA protein with the transmembrane domains. You're forming these clusters, and once you cause this conformation change, protons can move through this channel. And as the interior acidifies, you are going to start loosening the core and ready to release particles into the cytosol. Now, once you have these nucleocapsids, they need to enter the nucleus to make mRNA. This is where mRNA and RNA synthesis is going to take place. So RNA replication can take place. So here, again, influenza virus, getting endocytosed, you have your early endosome. It's also walking along these microtubules, and as the endosome matures, you are releasing the nucleocapsid close to the nucleus. Okay? And these will now get internalized into the nucleus, where you can make mRNA as well as replicate the genome. Okay? And the way it works is actually very interesting, because just by controlling the concentration of one of the proteins, you can switch between transcription or replication. Okay, so you either make mRNA or you make a full-length copy. So how does that work? So the nucleocapsid needs to enter the nucleus, where your mRNA and RNA replication, mRNA synthesis and RNA replication can take place. Now this protein, the NP protein that's shown here in green spheres, they actually wrap the RNA into this unusual twin helical conformation, as you can see here. They have a central loop right here, and this wraps as a helical conformation. Okay? Now, these nucleocapsids also have a trimer of RNA polymerase proteins, the proteins we saw earlier, PA, PB1, and PB2. The PB1 actually binds to the conserved sequences at the 5' and 3' end. So these are the regions that I showed you guys earlier on either end of the genome, okay, or either end of the genomic fragment. So these are about 12 to 13 nucleotide sequences that are highly self-complementary, and your RNA polymerase subunits are going to bind here. Now, your NP protein as well as your RNA polymerase proteins. So this green protein and this whole set of proteins, they contain what's called a nuclear localization signal, okay? What that means is that they will target the protein complex to the nucleus. So as a result, it gets inside the nucleus. So it interacts with cellular importance. Who knows what importance are? Yeah.
That's what helps you get into the nuclear pore complex.
Okay, so importance are proteins that bring material into the nucleus through the nuclear pore complex. Okay? So these importance are specific proteins. They will bind to right kind of targets that have the nuclear localization signal, and they'll bring it into the nucleus through the nuclear pore complex. Okay? And unlike other RNA viruses that we have seen before, your orthomyxoviruses, they replicate in the nucleus. And as a result, the machinery that's required for viral replication becomes fairly complicated. Because what needs to happen is your nucleocapsid needs to go in, it needs to be made into antigenome, and finally into genome. But to do this, you need to send mRNA out, synthesize proteins in the cytosol, bring those in, and then send the whole fragment that's fully assembled outside the nucleus. Okay? So it's a fairly complicated process. So what we are going to do is we are going through this step by step. Okay? But before we do that, let's look at importation of this into the nucleus. So just to show you how nuclear import works, so you have a protein or a cargo that has a nuclear localization signal. It's going to bind to a dimer of important B alpha and important beta. Okay? They go through the nuclear pore complex. They usually just interact with the filaments that are on this side. It basically goes in after a conformation change. Now, inside the nucleus, you have lots and lots of RAN-GTP. It's a GTPase. And once RAN-GTP associates with this, your cargo gets released, and your important alpha and beta are also released from this complex. The RAN-GTP stays bound, and another set of proteins called exportins also send this important material and RAN-GTP outside the nucleus. Okay? So this can be recycled, and as a result, once it's inside the cytosol, the RAN-GTP gets hydrolyzed to RAN-GDP. Important alpha and beta, they will complex again, and then they're free to go bind another cargo molecule. So that cargo has to have a nuclear localization signal. Okay? So that's the basic import and export of material of these proteins from the nucleus. All right. So the summary of the steps is you import the nucleocapsid into the nucleus. Then you carry out mRNA synthesis, okay, viral mRNA synthesis. Then viral mRNA needs to be sent out and needs to be translated in the cytosol. But once you translate that, you are making all these different proteins, okay? So you import those proteins back into the nucleus. So you have your early proteins and late proteins. So early proteins would be your NP and your RNA polymerase subunits. Late proteins in this case would be matrix and NS2. Okay? So you make your early proteins, you bring them back into the nucleus. Then you replicate to antigenome and then finally to genome. And once everything is assembled, you send it back out into the cytosol. Okay? So, major steps. What's the first step? Nucleocapsid goes inside the nucleus. Then what? So you synthesize mRNA, then what? You send it out for translation into the cytosol. What happens then? Early proteins are made, you import them back in. Then what? You replicate first to antigenome, then to genome. And while you are doing that, you're also making your M1 and NS2, which are your late proteins. Those come in, everything joins together, forms the complex, and you send the complex out into the cytosol. Okay? Now the question is, how do we control all this? Okay? So first thing we're going to look at is the replication. Or sorry, mRNA, synthesis of mRNA. So these RNAs, they do not have a cap. So how this virus works is, it actually steals the caps from the cellular RNAs. That's why the mechanism is called a cap stealing mechanism. Okay? So your capped ends of cellular pre-mRNAs are used as primers for synthesis of viral mRNAs. Because the viral transcription machinery cannot make mRNA on its own. So as a result, it uses the cellular pre-mRNAs. Your PB2 will recognize the cap on the pre-mRNAs in the cell. Okay? Then PB1 cuts at a specific site. It acts as a nuclease, okay, as well as a polymerase. So PB1 is both a nuclease as well as a polymerase. And what this does is, it cleaves the bound pre-mRNA at either an A or a G that's about 10 to 13 nucleotides away from the 5' cap. And it adds a complementary nucleotide. Okay? So you can see that here. Here's your A, and you add a complementary nucleotide in this case, which is U. And this capped fragment can now be used as a primer for the synthesis of viral mRNAs. So here's your PB1. PB2 is holding the cap from a cellular pre-mRNA, and PB1 will now basically continue on with elongation. Okay? With me so far? Yes or no? Yeah.
In the transcript, they will ensure there will be an A and an U in the sequence.
Yeah.
So it cleaves at the A or G. So
A or G, yes.
A or G, and it like adds that A or G to that push. Like they already know there's a U over there, right?
Yeah.
Okay, I guess.
So it's going to synthesize because it's attached to the cap, right? So that's used as a primer, and then cleaves at that nucleotide. Okay? All right. So let's go through this process step by step. So first is, one thing to keep in mind here is that the viral mRNAs that are synthesized using this method, they have a heterogeneous sequence for the first few nucleotides. Because each of these, they come from a pre-mRNA cap. And these enzymes don't care which pre-mRNA they take. Okay? So as a result, this first 13 nucleotides, 10 to 13 nucleotides are variable. Okay? So it's a heterogeneous sequence. Because the genome does not encode any viral capping enzymes, and the 5' end sequence of the fragment will be dependent on the cellular pre-mRNA that was used. Okay? And this cap stealing mechanism is used by other orthomyxoviruses as well. Okay? And you can actually block the replication of influenza virus by inhibiting cellular RNA synthesis. So for example, actinomycin D and amanitin can do that. Okay, problem is you are disrupting cellular RNA synthesis. So what that means is you're going to start affecting your own cells. So ideally, not a good treatment. Okay, yes, you can use this in the lab to study the effects, but not for treatment because it will be very difficult if you start to inhibit all the RNA synthesis in our cells. Okay? All right, continuing on. So each of these genomic RNA segments has a stretch of poly-U's, which you can see here. Okay? So this is again about 15 to 22 nucleotides from the 5' end of the viral genome. Okay? So again, remember this is a negative sense, so this is 3', this is 5', and your viral mRNAs, they terminate in poly A tail generated by the stuttering of the RNA polymerase. Okay? Because what's going to happen is, now you have PB2 attached to a cap from a pre-mRNA from the cell. PB1 will cut, and using this as a primer, you're going to start synthesizing the genomic RNA. Okay? PB1 will elongate. PB2 comes off the cap. It's now ready to go attached to another pre-mRNA. And as PB1 continues moving along, eventually it will reach that stretch of U's. Okay? And as it does that, it's going to start to stutter. Okay? So after reaching the poly-U, your RNA polymerase pauses and stutters, and it reads through the poly-U multiple times. So basically, it stutters and keeps repeating this region. Okay? And as it does that, you're adding a poly A tail to your mRNA. Now, PB1 will remain bound. So as it gets past the poly-U's, one of these will remain bound. Okay? And as a result, it doesn't undergo full transcription. It stops, the transcription stops here, and you make your mRNA. Okay? Everyone clear? I know it's a little bit complicated, but please go through this step by step, and you will be able to understand it. All right. The transcription of these fragments, it generates a set of 8 viral mRNAs. The 1 and 6 are exported directly to the cytosol or cytoplasm, and your segments 7 and 8 can actually undergo alternative splicing. And since the splicing machinery is present inside the nucleus, it has to happen inside the nucleus. How does it work? These fragments, they have consensus sequences that are recognized by the cellular splicing machinery. What's a consensus sequence? What's a consensus sequence? Sorry, say that again. They are the same, not identical. No, they are not identical. They are not the same. Consensus sequence is something, it's a conserved sequence, and this is something that you most often find in these regions. These are the nucleotides that you'll most often find. They are not identical. There might be slight variation. So think of this as an average of what nucleotides you might find in these regions. Okay? So here's your NS1, and here's your NS2. And what we see is that these splicing consensus sequences are recognized by the cellular splicing machinery, and as a result, it will splice and produce different set of proteins. So again, NS1, NS2, and M1, M2, okay? But keep this in mind that only a fraction of these mRNAs actually get spliced. So if you were to look at the ratio of the unspliced to spliced mRNAs, you almost have 9 times as much unspliced RNA as spliced RNA, okay? Your unspliced RNAs make M1 and NS1, which is fairly abundant, and your spliced RNAs create M2 and NS2, which are less abundant. Okay? Now the question becomes, how do we actually switch from transcription to replication? Okay? Because you're not just making mRNA, you also need to make a copy of the genome. And this is a very elegant mechanism that controls the switch based on concentrations of a specific protein. So remember, your genome replication is going to create a positive strand molecule. It's the antigenome. And as that antigenome is being synthesized, it's going to complex with NP protein. And this produces nucleocapsids with antigenomic RNA. There's no stuttering in this case, and it creates a full-length copy of the fragment, okay? And the mechanism behind this is actually fairly poorly understood. We don't know exactly how it bypasses that stuttering, okay? And this gets copied to genomic RNA. The antigenomic RNA will get copied to genomic RNA in a similar mechanism, okay? But what controls the switch is the amount of NP protein that's present, okay? So let's say here's your genomic RNA, okay, negative sense. You're going to use this as a primer, and you're going to make a copy. But as you're making the copy, the free NP that's present, it will start associating with the newly made copy, okay? So the amount of NP protein that's present in the nucleus actually controls what happens, either transcription or replication. Your NP is binding to the growing RNA chains, so as a result, the concentration of NP will drop inside the nucleus, okay? And this drop in NP triggers synthesis of more RNA, okay? So it will make more mRNA and make more NP. That gets imported into the nucleus, and the concentration of NP in the nucleus goes high again. And as it goes high, you switch again to replication, okay? So the possible mechanism is that NP does not bind to capped mRNA with cellular 5' sequences, but does bind to uncapped genomic RNA, okay? That makes sense, because otherwise, remember, nucleus is making lots and lots of mRNA, its own mRNA. So if all the NP started binding to those capped RNAs, what's going to happen? You're going to use up NP, and then you're going to have nothing left for your viral RNA. So the possible mechanism is that it only binds to uncapped genome RNA when you're replicating, okay? So as the concentration of NP drops, you synthesize mRNA to make more NP, you bring it into the cytosol, and you replicate again, okay? And you continue doing that until you have enough particles. Now, the nucleocapsids are then exported from the nucleus as they complex with your matrix protein M1 as well as NS2. The newly made M1 is again imported into the nucleus. It forms a complex with the newly made nucleocapsids. So here's your M1, NS2, and it's going to complex with newly made nucleocapsids, okay? Now this NS2 protein, just like we saw earlier, in this case, instead of nuclear import signal, there's a nuclear export signal. So this is sending stuff outside the nucleus now. This is recognized by another set of protein called exportins. So importins bring stuff in, exportins send stuff out of the nucleus, okay? And this allows for export of these complexes to the cytosol. And if you were to look at the mechanism behind this, it's basically more or less the reverse of the importing process, okay? So you have your nuclear export signal, your exportin will bind to the protein that has a nuclear export signal, again bound to RAN GTP. This whole complex gets sent out the nuclear pore complex. RAN gets hydrolyzed to GDP, and the complex comes off. Okay? And you can export it back in and recycle these proteins. Okay? So you bring the material in, synthesize what you need to, and then you export it outside of the nucleus. Everyone clear? Yes or no? Okay. Now, as you synthesize viral proteins, these enveloped proteins will transfer through ER and Golgi, and they start to assemble in the plasma membrane to direct the budding of these virions. Okay? So here, for example, you are synthesizing all your different proteins, and you're going to send some of these out into the plasma membrane like we've seen before. Okay? And as the nucleocapsids move along, they will get packaged into these vesicles. Okay? Your HA, NA, and M2 are directed to plasma membrane through your Golgi network. Okay? And they tend to accumulate in lipid rafts. Again, something that we have seen before. Okay? Your cytoplasmic tails of these proteins, so HA, NA, and M2, they interact with the M1 protein that's present on the nucleocapsid, so it basically holds the nucleocapsid in place. And lastly, one important aspect is that only one copy of each genome gets packaged, okay? Because there are viral proteins that recognize and interact with specific RNA sequences within the nucleocapsid. And as a result, it packages the virions, or it gets packaged as bundles into the virion, okay? Finally, as the whole complex reaches the plasma membrane, you can start to bud off, and at that point, you are going to acquire the envelope, and the virus will be now outside the cell and ready to go infect other cells, okay? Now, just to add on, your neuraminidase, it cleaves sialic acid, as well as, or sialic acid, which is the cellular receptor that binds to the viral HA protein, okay? Neuraminidase is a type 2 transmembrane protein, has a short cytoplasmic N-terminal, whereas you have a long C-terminal domain that extends outwards. So this is type 2, so this is the reverse of what we saw for type 1. So orientation is flipped, okay? So there is some evidence that indicates that M2 is actually involved in pinching of these budding virions from the plasma membrane. So again, let's have a look at the figure on the left. Here's your nucleocapsids, and they are going to start interacting with those NA and HA proteins that are at the plasma membrane surface. And as that happens, you're going to start budding off, okay? Now, the model indicates that the M2 protein causes depolarization of the membrane in that particular region where the vesicle needs to bud off, okay? So as a result, what ends up happening is your membranes start to fuse, and eventually you're going to release the mature virion outside the cell, okay? And the figure here shows you the same thing, more or less. Here's your neck and your transition. Here's your M2 protein causing depolarization of the membrane, and from there, the vesicle will pinch off, okay? Or the virion will pinch off. Everyone clear on that? Yes? Okay. Now, like I said, these virions, they do undergo fairly rapid changes. So part of the reason you have to get a flu shot every year is because we might get slightly different variant that's common in the population. So your influenza A viruses, they are divided into subtypes based on HA and NA proteins. There are 18 subtypes of HA called HA1 to HA18 and 11 different subtypes of NA protein. So again, NA1 to NA11. Most of these subtype combinations, they infect birds. Two of these subtypes, they circulate between human and birds. So, two of the common subtypes that circulate between humans is H1N1 and H3N2. H3N2 means HA3 and NA2. Okay. You also have two strains of influenza B that are included in seasonal vaccines. So your influenza A and influenza B strains are usually included in these seasonal vaccines. There's a very rare strain that may happen, which is H5N1, and this is bird flu or the avian influenza that rarely happens in humans, and it transfers from birds. It's not easily transmitted from human to human, but it can go from birds to humans. And one of the highly pathogenic versions, this is the H5N1, is the highly pathogenic avian influenza, okay? And if it does get into humans, the mortality rate is 60%. Almost reaches the mortality rate of Ebola viruses, okay? The influenza B viruses, they are not divided into subtypes. It's the A type that's divided into subtypes. Now, as these viruses mutate, there's a lot of genetic variability, okay? So it generates new strains that can potentially cause pandemics. How do these changes happen? Well, you can have antigenic change, which prevents lifetime immunity. And this can happen in two different ways. So antigenic change is basically changing the protein or the component that your immune system recognizes. So the antigen changes so that the immune system is not as effective. So how does that happen? Well, one is slow accumulation of mutations. So slow but continuous accumulation of point mutations. What are point mutations? What's a point mutation? Okay, one nucleotide change. Okay, so over time, the replication does make mistake, and you do tend to accumulate those point mutations. So that's antigenic drift. So over time, antigen is changing one nucleotide at a time. Then you get the antigenic shift, and this is more dangerous because it results from exchange of genes during a mixed infection. So reassortment of the fragments of two or more subtypes. But that means you must be infected with both subtypes at the same time. And as a result, it can potentially create a new subtype and a potential for pandemics, okay? So if you look at the top part first, this is antigenic drift. Let's say this is the antigen that we are trying to detect with the antibodies, okay? You have your various genome fragments, and you have your H and N antigen, or HA or NA proteins. Let's say one mutation happens, you have a slight change, okay? Might still be okay, but over time, if the mutations continue to happen, you're going to change the antigen such that it's not recognized by the antibodies anymore, okay? Or it's not recognized as efficiently, okay? That's one, that's your antigenic drift. But let's say you got infected with influenza virus from an animal, as well as a human influenza virus. So now you're mixing two different strains. But because the genome is fragmented, the fragments can get packaged differently. There's no way to tell the difference, okay? So you can mix and match these fragments from two different sets of strains. And as a result, you're going to create a new strain. And this strain, if it's deadly or if it's easily transmitted, it has the potential for pandemic. Okay? So for example, let's say there's avian H3 and whatever N number that might be. And we have a seasonal human H2N2. Okay? Now, through the antigenic shift and mixing of these two, you're going to generate H3N2, and this can cause pandemics. Okay? Now, this strain itself can undergo antigenic drift and become seasonal strain. Okay? So this strain that did not exist before, now you have mixed N2 from human strain and H3 from an avian strain. Okay? So as a result, you just created a new strain that could be potentially deadly. Okay, so again, here's human H2N2, avian H3N8. Let's say the pig gets infected. So you get genetic reassortment and antigenic shift, and now you're making H3N2. And these can undergo point mutations to give you antigenic drift. Okay? So these can continuously change, and they can change fairly rapidly. So a new pandemic of the influenza A strain, this arose by genetic shift, and it spread worldwide back in 2009. There were roughly about 20,000 reported deaths. And this then led to a massive vaccine development campaign. Okay? So again, you have your influenza virus, you have an epidemic strain that's transferring, then affecting the animals. Now you're mixing and matching these different fragments, resulting in a pandemic strain. Okay? You guys can look through the figure here, and it tells you where these different strains originated. Okay? Lastly, your vaccines are highly effective in preventing serious illness. There is a universal influenza immunization program in Ontario for individuals that are 6 months or older. But priority is given to high-risk groups first. So for 23-24 season, seniors are prioritized. Okay? So what populations are vaccinated and when can the vaccination start depends on a lot of factors. So the government has a set of criteria that needs to be met. So for example, people who are hospitalized and people who work in hospitals or long-term residents and caregivers, they are the first ones to get these vaccines because they are at the highest risk. And eventually we get back to general population. Okay? And one thing to add on, generally you need higher dose in seniors to get a better response. Okay? All right. So if you can explain all these steps, you know how influenza virus works. Okay? Everyone good? All right. So don't forget, no lecture next week. It's reading week. Your quiz 6 is available based on lecture 6. It'll be available on the Wednesday, due Friday, and not next week, the week after next, we'll start retroviruses and we'll talk about HIV. Okay? All right. See you guys next week. Well, in 2 weeks, sorry. Yeah.
So for influenza B, you mentioned it doesn't go by the subtypes like H number, N number of the spikes. So
That's the influenza B type, yeah.
yeah, so does that mean that it has does it have different spikes or is it always just one type of HA, one type of N?
Usually it's just one type.
Okay.